So another way to measure activities is to look at the metabolites, a lot of enzymes, right? So a lot of the enzymes, they will convert substrates into product. So if you can use metabolomics to measure, the substrate and the product formation, you're doing activity-based proteomics. So essentially combination metabolomics, proteomics. So these are really, really powerful because a lot of time protein changes may not correlate with activity, which is really important. And protein localization, trafficking. So this is a work I did during my postdoc at the University of Pennsylvania. So I was working on the yeast vacuolar proteome. The yeast vacuoles, they're very similar to human. It's kind of like a model for human lysosomes. So I isolate, really pure vacuoles and then run 2D gel, identify the proteins. But you can see from this gel, there are a lot of enzymes. They're supposed to be cytosolic, like indolase, phosphoglycerate kinase, all these enzymes. Of course, you have vacuolar ATPase, saccharopepsin, these aspartate protease, carboxypeptidase, protein disulfide isomerase. So these, they're in the vacuoles. But a lot of enzymes like fructose bisphosphatase, they're vacuolar proteins. So when my supervisor saw this, and he said the vacuoles are contaminated. So we cannot publish this. It's all contaminated. So the reason I show this is to highlight the power of genetics combining with proteomics. So in yeast, the cytosol to vacuole protein transport pathway is all figured out. So you have mutants you can use. So essentially you have mutants which is blocked in the cytosol to vacuole pathway. So then I took the mutant and I run the mutant as well, extract vacuoles, purify vacuoles from mutants, and do a comparative analysis. If the mutant, I don't see these proteins anymore, that means these proteins are bona fide vacuolar proteins. They're transported by that cytosol to vacuole pathway. And when I do the mutant comparison with wild type, suddenly this question becomes clear because in the mutant, I don't see these enzymes anymore. So that means these enzymes, these cytosolic proteins, are transported to vacuoles for recycling. So that's just a highlight, the trafficking. And also, especially people working with serum, human proteome, the biomarker discovery, using the body fluid, urine, serum, they're really good samples. But the problem with the serum, you have a lot of abundant proteins, dynamic range is so high. So in the lab, we actually use several approaches. One is the antibody-based depletion. So you can deplete these 20 most abundant proteins, then you start to see low abundant proteins. Another technology we use is from Biorade. It's called a proteome miner. So it's an enrichment procedure. So you can remove the high abundant proteins. You can enrich for low abundant proteins. So for those of us working with plants, rubisco, which is the enzyme fix carbon dioxide into sugars, that is the most abundant protein on Earth. So if you run these on 2D gels, you're going to see them. They disrupt this 2D gel run. So these are large subunits, this is small subunit. So we do have antibodies to remove this rubisco from our plants. So this is a technology I mentioned, proteome miner, is a really good. I think it's much better than antibody-based because when you do affinity-based isolation of these serums, you do remove some of the target, some of the biomarkers unspecifically, because those proteins are shown to bind to these IgGs or serum albumin. But this proteome miner, you can see, this is the same amount of loading. This proteome miner, they don't remove this serum, serum albumin completely, but they deplete them. In the meantime, they are enriched for low abundant proteins. So this proteome miner essentially is a hexapeptide bind to the, agarose beads, and this is a peptide library, so it's supposed to bind to all the proteins, but when you have high abundant proteins, you saturate those beads very quickly, so then the rest will be all washed away. So you kind of deplete these high abundant proteins. While low abundant proteins, you don't saturate those hexapeptide beads, so you can keep binding, then you get this enrichment. So this is a really good technology we use. You see this is manatee serum. If you do depletion using protein miner, you see a lot more spots on the gel. And also top down, as you know, a lot of times we don't capture the modifications. Tong has been working on this phosphorylation, phosphoproteome. We miss a lot of phosphorylation side. Some of them are so low abundant. Even we do affinity-based enrichment, we still don't capture them. So top down now start to become more and more popular. So people have been working with, many large scale, LLC mass spec on this top down. Essentially, you don't digest your protein. You just, introduce intact protein into the mass spec, and then you sequence. You get 100% coverage, sequence coverage. So you can see the top down proteomics is very powerful because you don't digest, so you don't lose the peptides. So also nowadays, people also look at protein degradation and turnover as a way to regulate protein amount. A lot of times we see protein going up and down in the cells. Sometimes we jump into conclusions. This is because of biosynthesis. This is because of translation. No, it may not. Yes, maybe, but you have to. You cannot overlook the protein turnover degradation. Degradation is another side, which can change protein levels. So people have, developed this isotope labeling using SALAC, then monitor the protein degradation turnover in the cells, in yeast cells, and also in plant cells. Protein complex, protein-protein interactions, that's another big topic in proteomics. So, even for a simple transcription of a gene in E. coli, it involves hundreds of proteins. So, nowadays, especially we have quantitative proteomics, we can ask a question, what proteins we interact with what, and what are the stoichiometries. Tong has another project looking at kinase substrate interactions, how kinase form a complex. So, affinity-based proteomics, protein-protein interactions, really challenging because a lot of these interactions, they're really weak, they're transient. You may think about cross-linking so that you can do in vivo cross-linking so you can capture the complex. Then you identify them. So in the lab, we have one PhD student doing some cross-linking using formaldehyde. And it hasn't worked yet, so hopefully it's going to work sooner or later. So this is what I already mentioned. PTM is a big part of proteomics nowadays, especially signaling regulation, MAP kinase, turn on and off, redox modifications. So in my lab, we look at redox modifications and phosphorylation. And also these modifications, they crosstalk. So Tong has a protein, he can tell you more about it. It's a MAP kinase 4. So when it's phosphorylated and oxidized, it does not lose activity. It starts to aggregate. So it's really, really interesting when you have this PTM crosstalk and how that links to protein turnover degradation. So there are many ways to do this. In our lab, we have enrichment for phosphopeptides. After enrichment, we see mostly phosphorylation. Without enrichment, we don't see phosphorylation. Data sharing, data standardization, HUPO, Human Proteome Organization, that's the leading organization defining the proteomics standards. And nowadays, if you want to publish in many journals, you have to deposit your data into the common data repository, like a Proteomics Exchange. That's the one we put a lot of data on there for publication. And also, where we do proteomics, We also have a lot of problems. In old days, I know when I started proteomics, we run 2D gels, we identify proteins. At that time, we use a MALDI-TOF, one spot, we see mostly one protein, which is easy, which is nice. Then you can see that spot is that protein. But I'm sure we miss a lot because nowadays when we run 2D gels, I cut out a tiny spot. I have tens, hundreds of protein from that spot, right? So that's create a problem. And even for this human proteome, so this paper published right after the human proteome. The good thing is all the data are in a centralized location. So people can reanalyze the data. And these guys essentially reanalyze the human proteome data. And they found out there are many things do not make sense. For example, they published, in certain tissues, they published proteins that do not belong to that tissue. So essentially, it's a false positive, right? When you have such a big data set, we had to be careful how stringent we can be and how relaxed we want to be. So I already showed you. I do have a 2-day workshop. I appreciate if you can take this back to your institution and help us advertise this. The hands-on workshop is about LCMS, attract labeling for quantitative proteomics. And our lab, we have many mass spec, many people. This is a recent picture. Essentially, our goal is to serve the scientific community. So we got samples from China, from Malaysia, from a lot of different places, Mexico. So we're a service facility. We give you free consultation. We follow up. We help writing papers. We help analyze the data. So this is the workshop from last year. You can see people from different places, mostly graduate students. We do have faculty also attended this workshop. So this year is April 28, 29, 2 days, 2 full days. People are actually doing hands-on protein instruction, labeling, then LCMS, then data analysis. So I have a flyer over there. So I think this is kind of a quick highlight about proteomics, why we love proteomics. I think I don't want to spend too much time just talking here. So I want to, maybe our panel members want to talk. Awesome. Yes.
So one of my big concerns with running the 2D gels and identifying spots, you kind of alluded to it, that now with the technology, if you envision about 70% of the genome expressed in any given cell type, that's what I guess came out of some of that study in Denmark, then you would anticipate with alternative splicing, never mind modifications, about 100,000 protein species per gel.
Mm-hmm.
On the best of these gels, maybe you can see resolve 6,000 to 7,000 spots,
Mm-hmm.
which would mean theoretically you're going to have 10 to 20 proteins per spot.
Right.
When
Right.
you do the analysis, you're going to be seeing the abundant proteins, because they're the
Right.
peaks that are going to emerge.
Right. Right.
So when you're looking at cell signaling proteins, these proteins are present typically at concentrations that are about even 100 to 1,000-fold lower
Mm-hmm.
than metabolic pathway enzymes. So
Mm-hmm.
I remember going to a lot of posters over the last 20 years, and most of the proteins that are listed when they do this kind of approach, they're abundant metabolic pathway enzymes, structural proteins, but rarely do you see protein kinases and G proteins. Yeah, so it always made me kind of concerned that I'm missing a lot of the more interesting proteins, the regulatory proteins, but on top of that, and you can sort of see it in your images, you see the spots kind of shifting. Every phosphorylation event that's stoichiometric results in and it's a summation of signals.
Right.
Right? So
Right.
you may have different species of phosphorylation in the same spot, but phosphorylate at different sites, but the sum still gives you the charge to mass ratio being the same. So you really don't even know what state of phosphorylation it is.